Okay, let the record reflect that the defendant is present along with counsel for the defendant, assistant state attorneys. Both sides ready to proceed?
Yes, sir.
Let's return the witness.
While we're doing that, Judge, can I make a humble request?
Yes, sir.
I realize we have a short day tomorrow, but we would request that the court inform the jury that it is due to your honor schedule, not the defense, so
I will
they don't hold it against us.
Did I tell them the reason why?
No, you didn't. No, you didn't, sir.
No. I will gladly do that.
Thank you, sir.
It also occurred to me that the court had discussed at some point saying something to the jury about witness Rodriguez and his not coming back, and I didn't know if you recalled that, but I think at some point we do.
At some point, we're going to need to do that, so y'all might want to consider crafting some language.
We have probably some further inquiry that we'd like to broach on that topic. We're discussing that amongst the team now, so.
You want me to explain to them about the half a day when they come in now? Because I may forget later on.
That's fine.
Okay. Let's return to jury. Okay, good afternoon, ladies and gentlemen of the jury. Did you heed all of my previous admonitions? Okay. Remember tomorrow, which is Wednesday, we will only work until noon or shortly thereafter. The reason for that is because I have a long-standing commitment for tomorrow at 1. And just to give you a brief explanation, it deals with the state trial court's budget, which I serve on the executive committee of the Trial Court Budget Commission, which is having a meeting here. They have a meeting the next day, which I will miss. But that meeting is very important for the well-being of my circuit. That's where we discuss the distribution of funds for the next year. So I just want to make sure that we are represented when the bargaining begins. Okay? Mr. Byers, you may continue.
Thank you, Your Honor. May it please the court. Mr. Ashton, good afternoon, ladies and gentlemen. Good afternoon, Mr. Eikelenboom.
Hello.
Okay, we're going to talk about DNA. Now, sir, can you tell the ladies and gentlemen of the jury, give them an example of what touch DNA is?
Well, touch DNA means you have all kinds of DNA. Everybody knows about blood, saliva, semen. Touch DNA is DNA which is transferred mainly through the hands. So if I touch an object, I will leave DNA.
Is there different types of touch DNA, or actually, let me rephrase that. That's a poorly asked question. What affects how much DNA when you use touch DNA is left?
Yeah, there are different factors which will decrease or increase the chance of obtaining a DNA profile. If I touch this table here very slightly, the chance of obtaining a DNA profile is very, very remote. The upper layer of my skin contains DNA cells or contains cells, skin cells. But these skin cells are dying cells. I actually have this in the presentation. I'm not sure.
Your Honor, may we, the witness has a PowerPoint presentation that would assist him as demonstrative aid. When we publish that?
You may, what's the exhibit number for the demonstrative exhibit? Attach the exhibit number.
27.
Okay, it's for demonstrative purposes only. You may.
I don't believe it would be an item in evidence.
It's not in evidence. You just need to have an identification number, Madam Clerk.
So, actually, it's on his. It's a disc. I can.
Okay.
Give you a
we just
disc.
need to reference it at some point. It's a dish, but we just need an ID number for it.
Yes.
Okay.
CI.
and just label it a demonstrative exhibit.
Go ahead and bring it up.
Okay. Just without the slides then.
No, you can bring up your PowerPoint presentation if it helps you. May we publish this to the ladies and gentlemen of the jury?
You may.
It's visible for you?
Hold on a second. We're going to make sure they can see. Can everyone see? Oh.
Yeah, it's a delay.
Oh, okay, great.
Okay, go ahead and explain what we're looking at here.
So, yeah, the first slide was the common types of DNA everybody knows about and hears about. The second slide is about skin cells, epithelial cells. And these epithelial cells are the covering cells of the organs. But mainly when we talk about touch DNA, we talk about, of course, skin cells from the hands. If we go to the...
So when you say epithelial cells, you're referring to skin cells.
Epithelial cells is every covering cell. That could also be the mucosa from the mouth, or the covering cells from the lips, or vagina cells. But for touch DNA, yeah, we mainly talk about skin cells from the hands.
Okay.
Where we are
and what are
interested in.
What are we looking at here?
In this slide, you can see the top layer of cells doesn't contain any nucleus. That is the part of the cell where we do get our DNA from. So that means that if I touch something, I will leave this top layer mainly, which is hardly containing no DNA material. And of course, in order to obtain a DNA profile, we need cell material or cells which contain the DNA. If you look a little bit lower below the top layer, you can see that there is, you see these kind of square bricks.
And that's this section here that I'm highlighting.
Yeah.
Okay.
So under the pink and under the blue ones, you see this section below that. And in the square, those are cells. In the squares, you see a small oval, round.
That would be this section here, the spiniest layer.
Oh, so you're signing on it. Yeah, but also in the granular layer, you see a round circle, and that's what we call the nucleus. And that nucleus, that contains the DNA which we need in order to obtain a DNA profile. So in order to get DNA from skin cells, you can see that if you only have a slight touch, the chance of obtaining a profile is very, very remote. If I apply more force, and of course that happens more often with violent crime than a casual contact where you just touch somebody on the shoulder or touch the doorbell, then the chance of obtaining a DNA profile increases. With blood, saliva, and semen, we all have presumptive tests. These are tests which gives us an indication that blood, saliva, or semen is present.
Would that be like a phenolphthalein test?
Yes, that's correct. Okay. But there are lots of tests like luminol or fate bias for saliva, or it's like phosphatesmo for enzymes which are present in semen. So you have all kinds of tests. The problem for the epithelial cells is that there's no test. So we get into a problem in detecting these cells. One possibility of detecting the cells is using an alternate light source. You see this on CSI all the time. They use a darkened room with the blue light. Well, that's what we call an alternate light source. And then if you wear orange goggles, then body fluids can fluoresce. And especially body fluids like saliva or semen or urine will fluoresce very well. A little bit less, or less than these three that I just mentioned, is the sweat. Sweat also fluoresces. If you will look under the armpits of a t-shirt, you can see that very well. But sweat from the hands are a problem, because there's not coming so much sweat from the hands. So if you touch someone or an object, there will be sweat there, of course, depending on conditions, like it's warm outside or I'm heavily sweating.
What about sweat from the face and the forehead? Does that show up fairly well?
Well, it depends. If I just touch it once, it's very difficult. So in a lot of times you don't see it if you if you use the alternate light source. But at least we always try first, also in this case, see if we see certain stains with the alternate light source. And if that is sweat, then that could give an indication that somebody touched the location and maybe left also skin cells there. So you're not detecting the skin cells then, but you're detecting, you try to find sweat. And then you assume, or you make a working hypothesis where you think, okay, where there's maybe sweat, there also could be skin cells. Well, if you don't have that, and in a lot of cases we don't find that, then you have an alternative way of taking samples, and that's why you use a working hypothesis. And you looked for locations where force was applied. If, for instance, you have a rape victim and the shirt was torn apart, then you can look at certain locations where you can see, okay, it is very likely, or there is support for hypothesis that this is the location where a perpetrator might have grabbed the shirt and torn it apart. So force was applied, and that also means that the chance of obtaining a profile is bigger. So that's not a possibility. Still, you don't know for sure that there's skin cells there, so you have to do the testing in order to find out whether or not you get a profile. Then the second problem which you encounter, it's difficult to find, but it's also very difficult to obtain a DNA profile. As said, these dying cells hardly give a profile. With blood, saliva, and semen, in more than 95% of the cases, you will get a profile. With the skin cells, that's different. It's far less, depending per laboratory, but well, I think 50% would be a high score if you make it as a laboratory. So
let me stop you there. How do you combat these problems? How does a forensic scientist actually find these types of cells and this types of DNA? What kind of tools are available for that?
Well, as I was going to explain, we form the working hypothesis, and then you can use a number of amount of methods in order to obtain cell material or DNA from those cells, okay, even though they're broken down so much.
And how would you go about that?
That's the next slide. The point I want to make here is that the type of contact force is very important in order to on the chance of obtaining a profile. And then we have three different types.
So let me see if I understand what you're saying is you end up developing a working hypothesis by taking a look at the scenario and the type of contact and the location where that contact would be. Is that a fair statement?
Yeah, that's correct.
Okay. And starting from there, what do you do next?
Well, this is going into, you don't want to see this about the three types of contact?
Sure, go ahead.
Okay. Because I think in the Netherlands, at least, we use three types of contacts. And then you have, due to wearing, which of course is very often people wear clothing for a longer time, prolonged time, and watch. And then you have the forceful grip, which is of course more related to crime scenes and crimes. And then the contact due to touching. Due to wearing, you can understand that there's not so much force applied, of course, when you're wearing your clothing, but there's a prolonged time, and these clothing or items are more or less rubbing against the skin for a prolonged time. So you can understand that over time, so the longer that takes, the more DNA will accumulate, and you get a profile. And then these are a couple of examples like knife handles, clothing, watches, which could give profiles. So this category is the highest category of chance of obtaining a profile. The second category is important for crimes, because if a suspect doesn't know the perpetrator, then or a victim doesn't know the perpetrator, then of course it would be interesting to know whether or not you could find DNA on locations where a perpetrator might have touched the victim. This is talking about forceful contact. So now I'm not only touching it, but I strangulate someone, or I grab somebody very hard by the arm, or by the shirt, or by the trousers. So I go through the topper layer of the skin cells, the dead skin cells, but then now I come into a layer of skin cells which contain DNA. And then the third one is, I already explained, just a slight touch, a doorbell, a pen in a bank. Those is the worst category of chance of getting a DNA profile. There are a couple of factors which are also important. Some people shed more DNA than other persons. It could be that somebody has a skin disease or somebody is just shedding a little bit more. So a good and a bad shedder means that some people who donated more cell material when they touch something, we call it a good shedder. You also have people which, if they touch something or they even apply force on something, you don't get a profile from. They just don't give enough cells with DNA in order to obtain a profile. As explained earlier, long and short contact, the longer the contact, the bigger the chance of a profile. Strong, weak, also explained. And of course, you can imagine if you have a very rough object, it's almost etching your skin, then of course, you will also go to the top layer of the skin cells and you will grab material from the skin which contains DNA. Of course, two separate parts is that a sticky material, for instance, we have a method in which we use tape to collect DNA from clothing. So sticky material, of course, will also help to collect DNA better than just a smooth object which touches something. And
okay. Let me stop you there because I want to try and tie it in a little bit to this case. So let's take a piece of duct tape, for example.
Yes.
If a person were to cut a piece of duct tape, would they have to grip the non-sticky side in conjunction with the sticky side to cut it?
Yeah, if you cut it with your bare hands, then you get two locations. Of course, the sticky side where you have one finger and the other side where we have the other finger.
What type of pressure would be required to cut duct tape?
Well, it's more than a slight touch.
Objection outside his area of expertise is what pressure it takes to cut duct tape.
Sustain.
Have you had to ever recover DNA from duct tape before?
That's correct.
Okay. And do you find in your experience that you can sometimes get DNA from the non-sticky side?
Well, you have different types of sampling. Mm-hmm. And with the duct tape, if we know a location where, or we have in the working hypothesis location where it was torn off, and you can see that every now and then because the tape is more or less worn out. It's like stretched. Mm-hmm. Then we take a sample, and from those locations we are able to obtain a DNA profile.
Okay, and which side of the duct tape would be the most prone to being able to retrieve a DNA profile from? The sticky side or the non-sticky side?
Well, the sticky side, together with the force, of course, is the best location, because then you not only have the force you apply, but also the sticky material, and especially with duct tape, which is known to be very strong adhesive. Yeah, you will collect more DNA, of course.
Now, given the hypothesis that the sticky side of duct tape was applied to someone's face, what would you expect to find in regards to DNA on that?
Objection to hypotheticals not involving all the facts of this case.
Overruled.
Yeah, we had several cases where duct tape was used in robberies and that kind of and homicides as well. And the duct tape or the tapes, because it was not only duct tape, were fit for DNA.
What kinds of cells would a face, if it were covering a mouth and a mouth area, would you expect to see? What kind of cell material gets on that tape?
So if you cover the mouth, then of course the skin around the mouth. But then, as I mentioned earlier, if you cover the lips, you can. Yeah, the lips are wet, and there are also epithelial cells there. And of those, you all heard about the buccal cells, which are used for reference sample. So there's a lot of DNA in the mouth area. So those buccal cells are also very good for obtaining a DNA profile.
Now, given the scenario and the facts of this case, have you formed an opinion as to whether if duct tape was applied to this child's face, given the fact that it would have been applied forcefully and the stickiness, do you have an opinion as to whether you think DNA could have been collected from this sample?
Objection, compound question, and there's no clarity as to what facts this witness knows about this case that counsel's asking about.
Overruled.
Go ahead and answer.
Yeah, if the tape was put on the mouth and then was removed, it's about the case, or it's just in common now. If you would put tape on a face, you would get. It's very likely you would get a DNA profile.
Objection, counsel. The answer is not responsive to the question. About, I could explain more at the bench. He indicated tearing it off, which is not the fact of this case.
Yeah, that's his answer.
Let me ask you it in this way: if you had duct tape placed over someone's face and mouth, and that person died and was placed in a wooded area, even with the elements of an outdoor area, would you still expect to find DNA on that duct tape?
So at the start, of course, if you put it on the face, you would expect quite a lot of DNA there.
Okay.
Of course, the circumstances of in which a sample is contained, and of course I'm talking about the whole duct tape now, if it is in bad conditions, then DNA could break down. But, well, I had it later in the presentation, but of course with the PCR techniques, which are very sensitive, you could expect still to find DNA.
Okay. Go to the section of your presentation, because I want to try and keep it as directed to the issues for the jury. So let's go straight to that portion of your presentation so you can explain to the ladies and gentlemen of the jury why you have that opinion. Yes.
Okay. So what we do with DNA investigation, we investigate certain locations on different chromosomes, and those chromosomes contain information in which you and I are different. So because of these differences, we can make the difference between certain donors of DNA material on a crime scene, for instance. We amplify DNA, so we don't completely clone a person, but we look at small locations. And if you look at this picture, we have these small locations in the nucleus, on the chromosomes, where we do DNA investigations on. And what we do is we amplify those small locations. So now we have, with standard conditions, millions of copies of these couple of DNA or fragments which are present in the sample. So you start out with taking a sample, for instance, from the tape. You cut out a piece, or the FBI did swap a location, and then we have very little DNA. So what we always do is we amplify the fragments of interest, and that's what you see over here. You get all these fragments and we amplify them. Normally that's done between the 28 and 30 cycli, which means we have 28 to 30 amplification steps of these small particles of DNA. And then at the end, if there was DNA present in your tube, this DNA is amplified. Is that answering your question here?
Yes, go on.
In order to make it a little bit more clear, is that we can, theoretically, one cell is enough to get a DNA profile, not with standard conditions. So let's say between 50 and 100 cells are enough under standard conditions to obtain a DNA profile. That means that you cannot see it. So in this picture you see a slide. Well, you don't see, You cannot see what's on the slide, but still, if you look under a microscope, you would see cells, and that would be more than enough to obtain a DNA profile from.
Now, let me stop you there because I want to stay on the duct tape for a moment. Now, were you made aware that there were 2 partial profiles on this duct tape, specifically the Q63 duct tape that's been admitted into evidence? Evidence 302. My question to you, there were 2 partial profiles, one on the non-sticky side and one on the sticky side. Take this hypothetical for a moment. If on the, and now I'm only speaking about the non-sticky side, if the non-sticky side was contaminated, is there a possibility that that contamination could override DNA that's already there?
If you have large amounts of DNA, then it is possible to that you don't see peaks or alleles, what we call them, from small amounts of DNA.
And did you have an opportunity to look at how much DNA was there from the contamination?
The contamination peaks were around 150 RVUs. I have that in explanation later, but that's the RVU is a measurement which DNA scientists use in order to get an indication about the amount of DNA. And 150 is not very high.
Okay. So is there the possibility that there could be lower DNA masked by that, or does that rule that out completely altogether?
No, it's never ruled out. And what you see is that there's some disbalance in some of the peaks, which can mean that DNA of another person is present. But this disbalance can also be there because of the low amounts of DNA.
Okay. Do you have an opinion on that at all, as to whether that's possible, likely, or anything like that?
Well, I would have investigated the evidence a little bit further.
Not responsive to the question.
Stay.
Okay. All right, let's go to the non-sticky, to the sticky side. Are you aware that there was a 17 allele identified at the D3 marker?
Yes.
Okay. And when I say marker, I'm referring to a location that is tested for DNA, correct?
Yes.
Okay. And a 17 allele, can you tell the ladies and gentlemen of the jury what that is?
Well, an allele could be described as an appearance on a certain locations, what already mentioned, the marker location on a chromosome. And the alleles are the appearances in which you and I differ. So, and that's written down in reports very often as numbers. And you and I have the different numbers. So that's what it more or less contains.
Now, do you show how much DNA, how many RFUs are at from that 17 allele?
I have it in the presentation.
Okay, can you bring that up? Okay, what are we looking at here? It looks like some kind of a heart monitor.
Yeah. On the picture, you see different peaks and the peaks, which has a number, or the amelogenin, which is the
sex chromosome.
Yeah, the location where you can see the sex type of the donor of the DNA.
Okay.
So the computer is analyzing the data and is assigning these data. And what happened is that there was an assignment with the 17 and with the X location on amelogenin.
Now, because these are at such low amounts, is there limited information that you can get from this?
Yes, of course. What we try to do is to obtain full profiles. So the more information, a normal peak profile or electropherogram contains much more peaks with much more numbers below them. And that would, of course, give much more information about the donor of such a profile than if you have only 1 or 2 alleles.
Do you have an opinion after examining this electropherogram whether this is what is called an artifact, or is it or can you determine whether it's human DNA?
It's a small amount, but looking at this electropherogram, that I would say it's caused by human DNA.
Okay. Now, have you ever had the experience in being able to extract DNA from maggots or flies?
Yes.
Explain to the ladies and gentlemen of the jury how that's done.
When the maggots feed on human material, then it's possible, of course, that in the digestive system, if they feed on, for instance, blood, then of course there's cell material in there which contains DNA. If they consume that and it's in their system, then it's not completely digested at the beginning. So when you dissolve or extract the DNA from this complete insect or from the larvae, then it is possible that you obtain a DNA profile.
Okay. And tell me what kind of effect chloroform would have on being able to extract DNA, or would it degrade DNA in any way?
No, one of the methods used for DNA extraction is the phenol chloroform extraction. It's not so often used in Europe, but it's quite often used in the United States. And in that method, DNA is extracted with the help of chloroform and phenol.
Okay, so chloroform would never be able to affect being able to actually extract DNA that may be there.
I would not say never, of course. If you put DNA in a bottle of chloroform, that wouldn't help the DNA. But it's not that if you apply a little bit of chloroform, which is of course a volatile substance, that your DNA would be broken down like you have with chloride. So that's the difference.
Okay. I have no further questions, Judge.
Cross-examination.
Sir, if you could just leave that up there for me. Sir, do you have a PhD?
No.
In fact, you're presently a student.
Overall.
You're presently a student at the University of Denver seeking your PhD. Is that correct?
Correct.
Under the supervision of 2 other professors who are experts in the area of touch DNA, correct?
They are not experts on the touch DNA.
Oh, what is their area of expertise?
They're in the biology department.
Well, I mean, I assumed that, but what is their area of expertise? They're your PhD advisors, right?
Yes.
What is their area of expertise?
Well, Dr. Phil Danielson does forensic DNA, and it's mitochondrial DNA, for instance.
Right. And you're under their tutelage trying to earn your PhD.
Yes.
Your laboratory that you've discussed, the one in Holland, the one that you became the director of, you and your wife started that, correct?
Correct.
Sort of a mom and pop situation, just The two of you, I'm sorry, that's an American expression. In other words, the two of you started it and you were the entire company, the two of you.
No, actually the IFS was started with my wife and a co-worker from the National Lab.
So,
And I joined in 2005.
okay, so three of you.
Yes.
Okay. And your laboratory is actually in a converted barn. Is that correct? Correct?
We bought a farm and we rebuilt the farm.
Right. And you converted and retrofitted it, the barn, in order to make it into a DNA lab. You have, secure facilities and all that kind of thing.
Yes, so
In the
we
barn.
changed everything into a normal DNA facility, which is accredited as well.
Right. Now, in 2008, approximately, you and your wife decided to expand your business to the United States, correct?
Yeah,
And this was a business decision because there's a great deal more work potential in the United States than there is in Holland, correct?
you have more homicides over here, yes.
And you would agree, would you not, that the exposure that your company that hasn't started yet will get in this case is going to be extremely helpful to the future of that business venture? Wouldn't you agree?
Well, I'm not so sure about that, but we already work in the United States, in more than 7 states, and we're very busy with cases, so we're not really waiting on cases. The cases have to wait till we are finished. Our turnover times are more than half a year. So, no, we don't really need a lot of more attention.
But your laboratory isn't even open yet, is it?
No, in the United States.
In the United States?
The cases which we work on in the United States, we still do that from the Netherlands.
Right. And when you open your laboratory in Colorado, in the United States, it would be really nice to have a lot of work, wouldn't it?
We already have a lot of work, so actually we're trying to downscale it a little bit.
I see. And you're, the more work you get, obviously the more money you and your wife make on the new project, correct? And that's why you opened it.
Well, we're not really working always for the money. We have cases which we did for free, and so we rather work for in the way that we can work like we want to work. So we want to be able to work the way we want to do. And if we business-like, if we wanted to expand, we could have expanded way much more than we did now. But the problem is, it's very difficult to get good DNA scientists which can do this contact DNA, which we do a lot, in the way we want it. So we choose to be not too big.
Do you do something that's different than other laboratories? Let me strike that and ask another question. How many laboratories are there in the United States that do the same thing that you do?
I think it's growing now. I know that Body Technology tries to do something about contact DNA. I know that the American New York laboratory started to do contact DNA, also use low copy number DNA. And also the laboratory in Denver is start using more sensitive methods like we use.
Now, Bode Laboratory, that's a large laboratory. That's not a laboratory in a barn. It's a large concern.
Laboratory, yes.
Okay. So there are a number of laboratories in the United States that could do exactly the same thing that you do.
No, I don't agree. I think we have more experience than the other labs.
Explain to the jury what you mean by that. I mean,
well, with this contact DNA and touch DNA, objects which were worn by victims, what happened is that I think the first time we started with that in the Netherlands was 1997. And in 2000, the United Kingdom, or before 2000, but in 2000 we started research on low copy number DNA. The low copy number DNA means that you have the amplification, but you do more amplification cycles than the standard methods use. In the United States, this method was, well, actually not used, I think, till recently. In 2002, we validated the method, and in the National Lab, I was working on high-profile cases, and I used it in every case which I couldn't not solve So if we don't have DNA results, and very often then, if there was contact between a victim and a perpetrator, what you start doing is looking for contact DNA. But in order to do this contact DNA, you needed low copy number, especially in that time, to work more sensitive. Well, that didn't happen in the United States. And therefore, I think in the United States, the main state laboratories used mainly DNA from blood, saliva, or semen. But at that time, because I worked all these difficult crimes, we got a lot of experience with this contact DNA.
Did you invent the protocol and process of low copy number DNA?
No.
Where was that invented?
I think the Forensic Science Society laboratory, which is currently closing, I think, but I think they started the experiments.
Where is that?
United Kingdom.
You use a particular, do you use a particular kit or protocol that you purchase from some location in order to do this low copy number DNA you're talking about?
Well, in principle, you can use every DNA kit because the method is only amplifying more than the standard number of amplification cycles. So you can do it with every kit. We use different kits, especially the last couple of years. Some new kits came on the market which are more sensitive.
And you buy those from Applied Biosystems Incorporated.
That's correct.
Which is an American company, correct?
That's correct.
Now, let's talk for a minute. You said you can do things other people can't. Explain, let me strike that. The only thing you do extra is you run additional runs of the, let me start back again. PCR is the process of replication of DNA, correct?
Correct.
Normally, you said, I think in one of your slides here, normally it's a run of 26 to 30 runs of this replication process.
28 to 30, yes.
Okay, 20. And the way that works is it takes, basically the first run takes one intact cell of DNA, one intact strand of DNA, and creates a copy. So it makes two.
Yes.
Right. Next run, the 2 becomes 4.
Yes.
4 becomes 8.
Yep.
8 becomes 16.
Yes.
And beyond that, my math skills fail me.
30 to 120.
Right, exactly. So by doing this 20, 22, 25 times, you make hundreds of thousands of copies of this DNA.
Idealistically, of course.
That's what, I'm sorry?
Idealistically, of course.
Idealistically, right.
The problem with forensic samples is very often they're not like in a hospital. They're more difficult. There's debris or dirt in there which can inhibit the reaction. So you have to take that in consideration. That's why I also say they already laboratory were able to get a profile from one cell, but forensically that's not really realistic.
Right. So theoretically you could get it from one cell, but
yes.
Realistically you can't.
You need more forensically.
More than that. All right. I'm just trying to figure out what is there in that process that any laboratory with the PCR equipment can't do.
Well, it's the whole process. As I explained when we started the presentation, is what happened in the 90s was that a lot of emphasis was laid down on the DNA, the technique, and it was under scrutiny, and you were the first person to get DNA admitted. So there was a lot of problems around the DNA itself.
Sure.
Later on, after the techniques were refined and it was accepted in court, the problem became, okay, we have this very sensitive technique with the amplification, but we don't have the stain. So we have cases which we know a perpetrator had contact with the victim, but we didn't find semen or saliva or blood from this possible perpetrator. And that's when we started with the touch DNA, because we know there was a violent crime, the victim was killed, and in Holland that often doesn't happen with knives or bats. We don't have so many guns. So in a lot of cases, there was contact between the victim and the suspect or the perpetrator. And we had to find this DNA. And then you come to the contact DNA.
Right. Now, you said that your methods were validated in 2002. Were these methods, or maybe I misunderstood you. I thought you said 2002.
I think that was in the national lab. So they did a validation study in 2000 already. And actually, when they were busy with the validation study, I used it in a high-profile case already.
Oh.
So from that time on, it was in the Netherlands. It was used in difficult cases, cold cases. I was project leader on cold cases. So yeah, I used it in a lot of my cases at that time.
My point, I guess the original question I asked was that the only additional step that you do is you just take additional runs of the PCR method to make additional copies. That's the,
Yeah.
is that correct?
No, I didn't answer your question correct, so
That's all right.
I apologize about it. No, the problem, the point I wanted to make was that with the sensitivity of the techniques, it is possible to get DNA. It became more important to find the DNA. And till the day of today, that's still the problem. In a lot of cases, it's certain that the perpetrator had contact with the victim was dragged, but they don't find the location. So at that time, in 2000, it became clear that the technique was very sensitive, but you had to find it. And if you find something, or you had a location where you think, oh, this is a very likely location where the perpetrator touched it, you still, by a normal sampling method, you were not able to get a profile. So you had to play with the way in order to take a profile. For instance, over here I know the swabbing of clothing is a common sampling method. Well, in Europe this is hardly used because the results are not so good when you use swabbing. So we use a cutting method where you really pinpoint and you cut out a piece of sample and you dissolve the sample in the tube. And that's where we had a lot of success with cases, that we were able to find the right pinpoint location of DNA and then extract the DNA from that. But then, of course, the problem there is that if you have a piece of clothing, it could be that you need, if you don't know where to sample, you can take thousands of samples. And of course, as a state laboratory, you always are bound by certain amounts. You cannot do 1,000 samples on one item. So that was a big problem also in the national lab. You have to take the right position.
Well, my question really had more to do with the actual testing of the DNA, not the sampling. So I wasn't asking about the sampling, but the idea of running the test itself, it's simply a matter of running the PCR amplification for additional cycles.
For a low copy number, yes.
That's the only difference.
Sorry.
In that part. Okay. Now, let's talk specifically about a couple of the areas you've talked about. Counsel asked you about finding DNA in insects. Have you ever tested a particular insect called Megacelia scalaris, a fruit fly, a very tiny fly, and successfully obtained DNA from that type of fly?
No.
You indicated in your testimony that those situations where getting DNA from an insect is successful are situations where the food, that is the blood or flesh, has been recently ingested. And that if it's recent, that sometimes you can still find some DNA that hasn't been digested yet by the insect, correct?
Yeah. Well, I think I have to adjust that a little bit because the case we worked on and where we got the result was a 5-year-old case. So it's the remains where we got DNA profiles from. Whether or not at what time the insect died and or the pupae, the leftovers of the cocon, how you call it? Cocoon, where the insect is coming out. If they contain DNA, I don't know. But it could be in different stages. It could be that the insect or the larvae was dying at a moment, for instance, by drying, and that the DNA stays in there then.
Okay. But you did say in your direct examination that it has to do with some recency between the consumption of the food and the test, because obviously, eventually, the DNA is digested by the fly and is no longer. Right. Okay.
If it's digested, of course, the digestion of DNA, the digestion of DNA would break down, and the further the digestion goes, the more the DNA breaks down. And we measure small pieces, but of course, also with UV light, for instance, if you break the chain, you cannot amplify it.
Yeah. So basically, even if you were able to obtain DNA from a fly as small as Megacillia scalaris, if it had been a month or more since it had consumed whatever human food it consumed, it would not most likely be available. It would have been digested.
If the fly consumed the blood or other cell material and then lived through and died at the end of the cycle, then the chance of DNA obtaining is of course small. If it died in between,
Right.
before the consumption or the complete breakdown of DNA, then that's what I assume where we got the profiles from, is that, okay, then the digestion is halted, and then it's a way where the DNA could survive.
Have you ever done any published research studies on environmental effects on DNA?
I myself didn't.
You yourself?
No.
What is the most destructive environmental effect? What has the most destructive environmental effect on DNA? Number one enemy of DNA.
Well, it's a combination, I think, but moisture and temperature is a combination of moisture and temperature is a really detrimental effect.
So a hot, wet environment is the worst thing for DNA.
Yes.
And also the presence of bacteria is also extremely destructive to DNA, is it not?
Yes.
Would you agree, sir, that a piece of duct tape, well, let's talk about the duct tape for a minute. If you place a piece of duct tape on a person's skin and rip it off, it is likely to bring with it not only the dead surface cells, but the living sublevel cells, correct?
That's correct.
That's the point you were making on direct, correct? But when DNA is placed on a surface of the skin and not violently removed, then the cells that are in contact with the tape are in fact the dead cells, in direct contact with the tape.
Well, the dead cells, of course, the covering cells. Well, I think that's correct.
Yeah, the dead cells are the ones that are up against the tape.
Yeah, of course, depends how much force is applied.
Right.
And then you, but yes.
Okay. When it's torn off, it may pull with it some live cells, but what's in contact is dead, correct?
Well, in order to tear the other cells off, yeah, it should be also into the deeper layer, I think.
I mean, assuming it's not torn off.
Yeah.
That's my point. Okay. And you would agree with me, would you not, that duct tape placed upon the face of a human being who is then placed in a semi-swampy, occasionally underwater, hot environment in Florida, in an area that is an erstwhile trash dump, and sits there for 6 months as every bit of skin cell decomposes off the body, leaving it completely skeletonized. You would agree with me, would you not, that the likelihood of finding any intact DNA on that tape is extremely remote?
Well, of course, the conditions which you describe are very bad for DNA. This is not a condition where you would have your DNA in. But of course, depending on different circumstances, whether or not you lose all of it, as explained, we only need a smaller number of a small amount of cells in order to get a DNA profile. So the question then is, okay, would you lose all cell material? And that's possible, but it's also possible that still enough cells would be present to obtain a DNA profile, especially when using low copy number DNA.
But if there's no DNA on, when you test a bone for DNA, you don't swab the outside of the bone, do you?
No, if you want to identify the person of the bone, you cut out a piece of the bone or the skull. That's the two.
Scope. There was no bones brought up.
Because you don't expect to find any intact, recognizable DNA on the outside of the bone because it decomposes. Isn't that the reason?
The reason is that
Overruled.
Go ahead.
The body mass on the outside of the bones, of course, during decomposition has more, the DNA is degrading much more there. So why we use the bone and the skull is that because the chance of obtaining DNA profile from those locations is much bigger than from, and I'm talking about decompositional bodies which are in late stages, so there's no good blood anymore, and the skin and flesh are coming off the bones. That's the reason why we use it. Still, there is DNA in those locations, so it is possible, but it is way more difficult to get it from those locations, like for skin or the decomposing flesh or blood, than it is from the bones.
And while you have indicated that you believe it may be possible, you would agree it is extremely unlikely.
It will be very difficult to obtain a profile.
All right, let's talk about this masking issue that you discussed with Mr. Baez. You indicated that contamination in a DNA profile can mask existing DNA.
Yes.
But it would only mask existing DNA at exactly the same loci, correct? So going by your chart here that you use from your PowerPoint, the only thing that this 17 allele would mask would be a smaller 17 allele, correct?
This is not the
No.
profile that we were discussing.
It's an example,
Correct.
Judge.
Overruled.
Correct?
So well, with the lower 17, it's like 55. No, that would not be very likely. I think it was the other profile, the contamination, where.
Do you have a slide of that
Yeah, of course.
profile? Put that up, and we'll talk about that.
So
Let me clear this.
this is a profile where
This
there's
one.
more information visible.
Let me ask you the question then. Let's look at the various loci that we see here. Taking this 15 loci here, the only thing that would mask would be another 15 loci, correct?
That's correct.
It wouldn't mask a 14 loci.
No.
The same thing with the 16. It would only mask another 16. It wouldn't mask a 17.
No.
And the same could be said for each one of these loci. It would only mask its identical loci, correct?
Correct.
So in order for this profile to mask the profile of someone else, that person's profile would have to match at every single loci.
Yeah, it depends. What you see with the 17, for instance, what I already said, it would not be very likely that this profile is covering a full profile. So you will not lose a lot of information. What it would mask is, of course, the locations where you have the detection already, which you just circled.
Right.
And it's not so likely that a full profile would have been covered here. What could have happened is like a 17, well, in this case, there's no 17, but like a 16 or 15, if you have a very partial profile caused by a very small donation of a other party, that could have been covered. So then you would not expect that this covers a full profile.
Right. There's no way this covers a full profile.
No.
This masks a full profile.
No.
The only thing it would mask would be identical alleles.
Lower, of course, you see some alleles on this plate which are not mentioned, which are not detected by the threshold or by the analysis software. So therefore, you don't know completely if it's all these alleles. And also, if you look at this, this would be a good profile to enhance with low copy number DNA. Then maybe there would be extra information. So yeah, we would use this as a start for the investigation. Say, okay, maybe we should do low copy number and see if we get more information there. Also, maybe with a third person. If you look behind the 15, there's a very small peak, but with low copy number, it's possible that you get a 16 there.
I'm sorry, you're talking about this?
The peak behind. No, that's in front.
I'm sorry.
That would be a stellar peak.
Hold on. Show me what you can tell us straight with yours on the bigger screen here. if you use your finger, no, the one right in front of you. If you use your finger, you can circle or illustrate what you're referring to.
Okay. Oh, sorry, I'm not so good at this. Now, behind the 15, you see a small peak. And of course,
When you say behind, that's where you're losing me. I'm not sure what you're talking about behind.
after, just if you have the 15, where,
Talking about, that didn't work. Hold on. Let me get rid of this.
sorry,
Okay, so just kind of put a little spot on what you're talking about.
yeah, maybe I should just put it there.
Is it where you have your, that little peak
Wrong
back
one,
there?
sorry. The second blue one, there's a small peak above that.
Well, there's a lot of small peaks in here, right?
Yes.
And you're saying that some of that could be noise.
Well, if you use the low copy number technique, it could be happening that you get information out of there. From this, it's correct, and the factory, I think, they use 50 as a cutoff for the analysis. Actually, the analysis is done by the computer mostly, and then later on, after the computer is finished analyzing, then a scientist comes and rechecks if all the data is correct or he agrees with that. So the data is correctly analyzed, but there's information there. If you look at these rounds, and these ones.
And what could those be?
So,
What could those be?
Those could be, I think if we use low copy number on these would go above the threshold and you would get information out of that.
What else could they be? If it's not DNA, what is
If
it?
not DNA. Well, these peaks can also be artifacts.
Okay. And again, your comments about what low copy number DNA could possibly do are completely speculative. You don't know whether it would come up with an additional profile or not.
Well, I'm
Isn't that
pretty
right?
sure recognizing this location. The peaks we see over here in the two circles are on locations where normally also markers are. So
And
it's not pure speculation. That's that's the experience you need when looking over these profiles. Is that you recognize locations where you could get DNA extra information if you would use the low copy number method.
And every laboratory validates its equipment with certain limitations as to what will be called as an actual DNA peak versus what will not, correct?
Yeah, what I said here, it's the 50-hour view is a standard from the factory.
What is your RFU limits at your laboratory? Same?
We use the same.
So even in your laboratory, you would not call that as a DNA peak?
No, we would
Right.
not mention this in the report, but we would enhance it.
You would potentially run a couple more runs of
Yes.
PCR,
Yes.
and then if it still didn't show anything, you would call it as nothing.
No, if the computer and the analysis software, which is highly sophisticated, don't mention it, no, we're not going to mention it.
Did you ask to retest this tape?
Yeah, we mentioned that we could investigate a lot of items.
But
Unregistered tape, yes.
did you ask specifically to retest this tape?
Yes, we could retest the tape, yes.
It's not what I asked you. Listen carefully. Did you ask to retest this tape? Specifically this piece of tape?
To José Baez,
To anyone?
anyone. Well, we mentioned that we could investigate this tape, yes. This piece of tape.
Are you aware if items were, at the defense's request, sent for additional DNA testing?
Object outside of the scope and outside of this witness's knowledge.
Overruled.
Do you know?
Well, yesterday during the deposition.
Well, other
you
than what I
mentioned-
told you,
Yeah.
because you can't really quote me,
Oh, okay.
do you know?
Say move to strike.
I just told him not to quote.
Objection overruled.
Okay. So aside from something I may have told you at deposition yesterday, as of yesterday, you have no idea if any item in this case was retested at the request of the defense.
No.
No further questions.
Redirect.
Mr. Eikelenboom, you were asked about your laboratory being on a farm. Do you
Yes.
recall those questions? Your laboratory is actually known internationally as the crime farm, is it not?
Objection, Your Honor, as to what other people may call something.
Overruled.
Could I approach?
No.
Correct.
Okay. In fact, you've been sought out by other different countries to have DNA tested at your crime farm.
Objection, relevance.
Overruled.
That's correct.
And Mr. Ashton asked you about exposure. Your exposure, you've already had exposure internationally from your laboratory, have you not?
Objection, leading and relevance.
As to leading, sustain.
Have you had any previous exposure just in the United States while you were in Holland prior to you being involved in this case?
Yes.
What kind of exposure have you received?
Objection, may we approach?
You may. Objection overruled.
Mr. Eikelenboom, I want to talk about the recognition and exposure that you received prior to ever being involved in this case. When did you first start getting recognition from the United States?
Objection, it's not the question the court authorized him to ask.
Overruled. He can answer the question.
I can mention the name of the case. The first case was the Tim Masters case, which was an exoneration. We started with that case in 2006.
Okay, let me stop you there. What were you asked to do in that case?
We were asked to do contact DNA in that case.
And what do you mean by an exoneration case?
We were approached by a police officer who worked on a case, and she came to the Netherlands, or she was in the Netherlands on a visit, and asked, she visited us and talked about this master's case, and she asked if we would be able and willing to do DNA in this case.
And was this to help free him from prison?
Sorry?
Was this to help free Mr. Masters from prison?
That's correct.
And what happened? What did you do?
We came over to the United States, talked with the defense team. Because there was no money at that time, we evaluated the case. I worked on quite some miscarriage of justice in the Netherlands and thought, well, this could be a miscarriage of justice. So we decided to continue with the case. So, okay, we will, if the courts allows us, do contact DNA in this case. And I testified about contact DNA in the case, and the judge allowed the evidence to go to the Netherlands.
And the evidence came to the Netherlands, and what did you do there?
We did do the DNA investigation, trace recovery, and DNA profiling. And then we were able to obtain a DNA profile,
Okay.
which was not matching the person in jail, Tim Masters. We obtained a lot of DNA profiles, all not matching Tim Masters.
Okay.
In 2006, there was not much interest, or there was not a lot of media attention, but by the end of 2008, we were able to get a match with a suspect, or a former suspect in the case more than 20 years ago. And that was the time that they released Tim Masters, and it was the first miscarriage of justice, which was acknowledged in the state of Colorado. And by that time, there was a lot of media attention.
Okay. And you received international media attention as a result of that?
Yes.
And that was from being able to obtain a DNA profile from 20 years past on Touch DNA?
Correct.
Okay. And this person was found outdoors?
Yes.
And the clothing that you tested was subjected to numerous elements?
Yes.
Okay. And held for 20 years?
Yes.
And then you finally were able to find something that wasn't found before?
Correct.
Okay. And subsequent to that, have you received other exposure from other cases?
Objection, leading.
Question is not completed yet. Overruled.
Have you received other exposure from other cases in which you have worked?
Yes. Because of this case, we got other cases from the United States, which also get some exposure in national media here.
Okay. And did law enforcement agencies, as a result of that, travel to the Netherlands to consult with you?
Correct.
Okay. And were you able to assist them in their cases?
Yeah, we were currently working on several cases.
Okay. So this case is not the first high-profile case you've ever worked?
No.
Okay. Now, when you talked about running extra cycles and that's the only thing you do, there are actually kits that are sensitive, that are more sensitive than your standard kits when running DNA, is there not?
That's correct. I had it in the presentation, but shortcut it.
Sorry, I didn't mean. I'm trying to keep it as simple as possible. Now, when we say kits, we're referring to the type of what is used to try and generate the profile, correct?
Yeah, you could say these are the chemicals used in order to, of course, it's a very complex procedure, but it's chemicals used to identify the different locations on the different chromosomes and make a profile.
Okay, and one example of this would be the mini filer
Yes.
kit.
The mini filer is specially made for broken down, deteriorated DNA, so that could have been helpful in this case.
And it's not just running extra cycles. There's also possibly extending the injection time.
Correct.
Okay, and that would, there are basically techniques that you use that might amplify the DNA or make it a little bit clearer so that the computer reads it. Is that correct? That's a poorly asked question, I realize, but I'm trying to.
Well, I think it comes back to what I already described. The trace recovery is very important. And that was underestimated in the 90s. Everybody was looking at DNA. But if I have a big stain and I just cut it out and there's so much oil or grease in it, or especially metals with the PCR, it could happen that there's a lot of DNA there, but you still don't get a profile. So you need to have a lot of knowledge about the amount from such a stain. You cannot just say, I cut out 10 by 10 centimeters of stain. So cutting it at exact right location with the exact amounts of pollution, and also with DNA extraction, which I didn't talk about here as well, but DNA extraction, what kind of method do you use? The phenylchloroform will not be able to perform as good as a cleaning action as, for instance, the QIAGEN material. So certain filters, we use filters to clean the DNA, that would also make a difference in the chance of obtaining a profile. So it's the whole process from finding the stain till the end, or till analysis, in order to gain success with the DNA.
Now, and another reason that you are able to generate profiles where other labs do not. Also,
assumes a fact which is not in evidence.
Sustain.
Move to strike.
another thing that you
Just
do,
a second.
sorry.
Motion to strike granted.
Another thing that you do differently is, or another consideration is also the type of machine used to run a profile, correct?
That's correct.
Now, I know you have a slide for that. Can you bring that up and explain why that might help you generate profiles that other machines do not?
DNA is, can everybody see this?
Mr.
Yes,
Pryor?
yes,
Oh,
may
sorry.
we, Judge?
That's the same demonstrative exhibit number what, Madam Clerk? Okay.
So we see two machines over here. The one is slightly bigger than the other one.
They look the same size to me. Which one's bigger?
The right one is bigger.
Okay.
That will become visible if we look more closely. This is the 310, which we use. Is a smaller machine, and it's just one capillary.
And that's this line right here, correct?
That's correct. There's a
Let me just
line,
clear that out.
and actually this is a very small tube in which the DNA is separated, and then a laser detects fluorescent labels which we put on the small pieces of DNA, which we saw with the PCR amplification, And when the laser is hitting the label, it will fluoresce in different colors if it's a different mark or different location on the DNA. If you go to the bigger machines, they have 16 capillaries there. So now the laser has to go to 16 capillaries.
And that's these
The right
things
one
right here, right?
So the ABI 310, you can see it in the text, and the ABI 300 are different in the way that the bigger machine has more capillaries, and that means that at the same time, where we do one sample, they can do 16. Well, one can imagine that for bigger laboratories, the state labs and the national labs, of course, they have lots of cases. And
Objection, Your Honor, this is not within the scope of cross.
It is actually, Judge. Mr. Ashton discussed that the only thing that he does different is run extra cycles. I'm pointing out that there are significant differences.
so the choosing of the machine is also of importance in order to get a profile. And we really tested this thoroughly because we do a lot of counter expertise in the Netherlands. So we test laboratory, or we do counter expertise on laboratory, which use the, all the bigger laboratories in Holland use the ABI 3100, so the one with 16 or even more now. And we found out that extracts where those laboratories actually didn't get any result, we were able sometimes to get full profiles, partial profiles, but we get a lot of results. And the only thing different, because we're using the same extract because they made the extract, so the only difference in the whole method was the machine. So that's why how we found out that the machine makes a difference as well. And of course, it does make sense if the laser has to go to one capillary. This is a glass capillary. If it has to go to one, of course, it's more sensitive than it has to go to 16, because you can lose some power of this laser light going through. So the machine using that it's slow, so that takes time, and that's why I say we have a turnover time, which is longer, because we don't do 16 samples at the same time. We do one sample at a time.
Now, Mr. Ashton questioned you about degradation of DNA and how water and heat are bad for DNA. Do you recall that line of questioning?
Yes.
Have you found DNA profiles, and do you have any examples of what you can show the jury of profiles that items that you've been able to obtain profiles in that have been exposed to these types of elements?
Do you want to see the slide?
Sure.
Yeah, in this slide you see three different kinds of pieces of evidence. You see a panties, which is more than a panties, pantyhose.
Yes.
On the left side, left upper side, and that's a piece of panty which is more than 20 years old. And after 20 years, we were still able to get contact DNA from these panties.
Now, I'm a little confused because this looks like there's a lot of blood here. How do you know? I mean, isn't that just the blood the DNA profile that you got from the blood, or is that?
The blood was from the victim, and of course, and it's more or less a given in the homicides we work on, is that you always have a lot of DNA of the victim, and of course, during our investigations, we're not looking for DNA of the victim, especially not on the clothing of the victim. So we're looking for DNA of possible perpetrators, and in this case. Of course, and that's why the sampling is so important. If you take a stain straight into the middle of the blood stain, you will have an enormous amount of DNA of the victim. But what you need is the DNA of a possible perpetrator. So taking a sample on a location, for instance, where there's less blood, where you're able to still enhance a profile of a second donor, that will help in order to get a profile. And that's what happened in this case.
Okay. And what else do we have here?
The second is the right hand of a victim which has been lying almost 3 weeks in the water. And also in this case, we were able not only to detect DNA from the victim, which if of course also 3 weeks in the water is not a very good condition, but also DNA from a suspect, which was later convicted, based also on the DNA.
Okay, and what about here, the jacket?
And the jacket is more or less the same. This jacket was not a body, but it was worn by a victim, which was lying in the water for around 3 or 4 days. And we were still able to obtain DNA from a suspect there.
So while, if I understand you correctly, while these conditions certainly don't help,
No.
they don't preclude you from being able to obtain a profile. Is that correct?
That's correct. Very often the bigger labs or the labs say, well, this has been in the water, we don't investigate it. And that's where we say, okay, well, give the evidence to us, we will try to investigate it. And then we were able to get DNA from that.
Okay. Now, you had requested to do DNA on items in this case, did you not? You were willing and able to do it?
Yes.
And you were willing and able to do it pro bono?
Relevance.
Approach.
Judge, just to move it along. Yeah. And the only reason you did not do the DNA testing in this case is because the prosecution objected to taking it.
Your
didn't you?
Honor. Relevance. Yes, please.
Okay, we'll take our mid-afternoon recess to 20 minutes past the hour of 3. Please remember all of my previous admonitions. You may be seated. Okay, objection will be sustained in the sense that you can question him about the question that Mr. Ashton asked about, did he ask to examine that piece of duct tape? I guess that was on the mouth.
Allegedly, sir.
Well, wherever it was.
Yes.
The Q62, 64 group of items, yes, sir.
And as to the other issue that you raised at the sidebar about asking the court to take judicial notice of different testing, give me exactly what you want, because I am really going to need to research that because of the issue of burden shifting. There is a very delicate and fine line with that. And that's something that I don't want to have to deal with at the last minute, because if I get it at the last minute, I'm going to err on the side of being safe and disallow it. But I need plenty of time, because those cases are all over the board, and it depends on which group you have, and the cases have shifted from time to time, just as the composition of the Florida Supreme Court, or if you look at the District Court of Appeals, whichever panel you happen to get. So that is not a very easy legal issue to decipher. Some things are best left unsaid. We'll be in recess.
Yes, sir.
Okay, state recognized presence of the jury and the defense.
Yes, sir, we do.
You may continue.
Mr. Eikelenboom, the 17 allele that was found on the sticky side of the duct tape, it was found at one marker, correct? The D3 marker?
Yes, 17.
Okay. And in order to include someone as a match, you have to have all 13 markers identifying that person, correct? For inclusion, I guess, generally speaking now.
No, what you normally do is you have a reference profile, and that's in the United States, it uses 13 core loci, profiler, co-filer. And ideally, you would have a match on all those loci. Okay. With a partial profile or a very partial profile, as in this case, you can only compare a reference profile, complete reference profile, with the 17 or the locus D3 here.
Now,
And the amylogenin.
Now, you testified earlier that you believe that this was human DNA, correct?
Yes.
17? Okay. Now, for exclusion, to exclude someone, how many markers do you need that is different from that person's profile? Did I ask the question?
As I understand correct, if somebody doesn't have the 17 in his profile, of course, if you don't have the 17 in the row of numbers of the 13 core loci, well, then you cannot have donated the 17. Is that what you mean?
Correct. So if I am a 14, 15
Yes.
at this marker, which is the D3, and a 17 shows up, could you exclude this person?
Yeah, you would not be the person who could have donated 17 then, because it's not in your profile.
Okay. I have no further questions.
Recross.
Ross.
Thank you, sir. Sir, one of the challenges with, let me restart. Original DNA testing was done with a system called restriction fragment length polymorphism, correct?
Correct.
And that system required a fairly significant amount of genetic material in order to get a profile.
Correct.
Sort of a quarter-size spot of blood or a bigger
Yes.
or a substantial amount of semen.
Yes, correct. For blood and semen mainly.
With advances over through the early 90s, this PCR approach became the approach used by most laboratories, correct?
That's correct.
Because it replicates even a small amount of DNA to make it larger, to make it visible.
Correct.
As that has happened, the concern for contamination has become greater.
Correct.
In fact, the smaller amount of DNA you're looking at, the more difficult it is to relate it to the crime itself. Isn't that true?
That's correct.
So in other words, that you might find a speck of DNA through even just regular PCR or low copy number doesn't necessarily mean it relates to the crime.
No.
So the fear for contamination is much greater.
Correct.
Now, in this particular case, and I'm going to... I think Mike will be able to pick me up. If you had tested the tape in this case, there are 3 possible results that you might have achieved. The first is that you might have found nothing. Correct?
Yep.
And if you found nothing, that would not be inconsistent with the tape being placed on the child prior to decomposition, would it? Because as you said, the DNA could have simply degraded and decomposed, correct?
That is possible.
Yeah. The other possible result is that you might have found Caylee's DNA on the tape, correct?
Correct.
And that obviously would be pretty strong evidence that the tape was placed over her mouth before she decomposed, correct?
Correct.
The third option is you could found an unknown DNA profile on the duct tape. Isn't that correct?
Correct.
And there would be absolutely no way to ever know whether that unknown profile had anything to do with the crime. Isn't that correct?
Well, I think the district attorney was hypothesis, what we call it working hypothesis, was that the tape was crime related. So what you do with the DNA testing is test whether or not you get a result, and you can substantiate or refute, find support or no support for a certain hypothesis.
But my point is, if you got a result, an unknown result, a result that didn't match anyone that anyone knew anything about, you would never know whether that DNA came from something related to the crime or some contamination, which is very, very possible with low copy number DNA. Isn't that true?
Okay, low copy number DNA is more sensitive or prone to contaminations. That's true. We see that in the case we work in the Tim Masters case, for instance, we had a lot of contamination, which makes the whole investigation even more difficult. Having said that, What we do now is, and that's actually what the FBI also did, is testing that against all the persons in which you know who worked on the case, the crime scene investigators, lab technicians, everybody had contact with the evidence. You have a profile from. And I think in the United States also you have this database of profiles, not only from suspect, but also from lab co-workers. At least in Holland we have that. So everyone working on crime scenes or working in a laboratory has to give his profile and will be compared to obtained profiles. So if you can exclude, or there's no support for hypothesis that any of these co-workers contaminated the crime scene or the evidence, then of course it becomes more likely that it is in some way crime related.
Well, but because this test is so sensitive, you can't ever know whether somebody had contact with their wife and then got transferred DNA onto an item because it's so hypersensitive. You just can't ever really know. Isn't that true?
Well,
you never really know.
No, it was low copy number. It's an issue which is more common. Of course, the type of transfer you're describing now is a lab technician having contact with his wife and contaminating the DNA with the DNA of his wife, but then still you would expect more DNA of this lab technician than from his wife. So this is called secondary transfer. Right. It's all possible. I won't deny that's possible. Secondary transfer is described, and there actually is literature about it. But it is less likely than direct contact, and so contaminations are easier found, and we, in the case we work on, we regularly find contamination by police officers or lab technicians. But then we recognize those.
Of course, the problem with low copy number is you may find unknown DNA, and then you'll never know whether it's contamination or an unknown suspect. That's the confusing part.
It's possible that it is that you don't find out.
In fact, that happens fairly frequently, where you get an unknown profile with low copy number DNA, and you really don't know where it came from.
That's possible.
Could you pull up that slide that you pulled up with Mr. Baez, the one with the jacket and all that in it?
As a part of the demonstrative exhibit by the defense, number what, Madam Clerk?
CI. Thank you, sir. The first object in the top left, was that associated with skeletonized remains?
No.
The object to the right, that obviously is a human hand that is not fully skeletonized. There's still tissue there. Is that correct?
Correct.
And the jacket that we see here, that was that associated with fully skeletonized remains?
No.
No further questions? Oh, I'm sorry. I don't think that, I've just told that may not have been published, and that's my fault. Let me go through that again. I apologize, just so the jury knows what we're talking about. May I publish it? Okay. None of these three, just to sum up,
Yep.
were associated with fully skeletonized remains, were they?
That's correct.
No further questions?
Redirect.
Based on what you do, these issues, the fact that they were, whether it's skeletonized remains or not, have no bearing on whether you would get DNA or not, correct? An item is an item.
Yeah, if there's DNA in the item.
Like, for example, these panties, you're not testing the skeletons, you're testing the item.
Correct.
Okay. Just like duct tape is an item.
Yes, correct.
The fact that the remains are skeletonized have no bearing on the item, do they?
Well, of course, it can give problems, but the item itself, it can be investigated. So, and then the condition of the item is more important than the skeletonization of the body. So the condition of the item, of course, is important.
And Mr. Ashton's three scenarios also leave out a fourth important scenario, do they not? Number 4 could also be that this duct tape was not wrapped around Caylee's mouth. Is that not true?
That's correct.
And in fact, based on what you've testified already, you would have likely seen some DNA based on the scenario that the state presents, does it not?
Well, if there was DNA, if the tape was placed over the nose and mouth, and especially the mouth and the mucosa from the nose, then there would be, at least at the beginning, there would be DNA.
And Mr. Ashton asked you about contamination and how sensitive DNA is now. When you find the perpetrator's DNA on an item, and you've testified in your experience that that DNA profile matches the perpetrator or the suspect.
The suspect and
Yes.
later convicted persons.
You have never gone into court and said, you just can't ever know.
In what sense?
Well, Mr. Ashton was questioning you about, you just can't ever know if if someone's DNA is on an item relates to the crime.
Well, the final decision whether it's crime related is in our system up to the judge and in this system to the jury, of course. So no, we would never go further than there's support for hypothesis this DNA came from this person, and on activity level, so it could be transferred. There's support for the hypothesis it is transferred during the crime or during pulling down the panties, for instance. So that's where we would go with this evidence.
Right, the evidence is either there or it's not. You don't give any interpretations and say things like, you just can't ever know.
No, it's on a crime level, yeah, it just match or doesn't match, or it would support a certain hypothesis or it wouldn't support a hypothesis.
There's either DNA there or it's not. Correct? I'm speaking generally, I'm not.
Yeah, well, if you have alleles, there's DNA, and if you don't, then there's no.
No further questions.
Just one. Are you telling this jury that the proximity of tape to rotting flesh does not increase the likelihood that the DNA on the tape would decompose as well?
It depends, of course. If the tape was on the body, which is decomposing, that would increase the chance that the DNA on the tape would also decompose.
No further questions. Thank you. Oh, I'm sorry. I thought you were done. Apologize. Go ahead.
Of course, it's depending if it's not on the body or it's a little bit away from that, then of course the chance of degradation of DNA is smaller. Of course, the outside conditions are still bad for DNA. On the other hand, of course, we have the PCR methods, which is so sensitive. We only need a couple of cells to obtain alleles or profiles.
So it does make a difference if the tape or item is in contact with decomposing flesh. It does make a difference, doesn't it?
Yeah, in my opinion, it does.
No further questions.
Yeah. Okay, may the doctor be excused for today. Thank you, doctor. You may stand down.